Okay. Okay. Well, so thanks to everybody for joining us for what I think is the last presentation for this year in our CCPBioSim Industry series. But please stay tuned because we have more presentation coming in 2024 on diverse topics. So we have quite a few more presentations lined up. And today, it's really a great pleasure to welcome Peter Kenny to give a presentation about hydrogen bond donor acceptor asymmetries in drug design. And before Peter starts, I'll say a few words about his background. So Peter is currently retired, or he pretends to be, because he's still working, in fact, apparently as a scientist, including giving presentations to strangers. So Peter was born in Port of Spain in Trinidad and Tobago and studied chemistry at Reading and Oxford universities. And then following this, apparently he got a lectureship at University of the West Indies, but then did a postdoctoral studies at University of Minnesota. And then he switched to industry. And he has a very long and distinguished career in industry. So he first joined ICI and stayed with this company through first the demerger to Zeneca, and then the merger to AstraZeneca. So you can see that Peter has done quite a few things in industry, and his interests, and he will talk about that today, include hydrogen bonding, alkane water partition coefficients, and aqueous solvation. And as I said, Peter now pretends to be retired from AstraZeneca. And since his retirement, he has collaborated with Brazilian scientists in the search for new treatment for parasitic diseases. He has really a great depth of experience in drug discovery, industrial work, and also academia. So the floor is yours, Peter.
Oh, well, great. Thanks very much for the intro. And thanks very much to everyone for sort of coming along. And I really appreciate the opportunity to present today. So let's get cracking. What I'll be talking about, first of all, is I'll take you through some drug design basics. I'm going to talk about polarity and hydrogen bonding. I'm going to show you how you can estimate polarity both in a molecular sense and an atomic sense from measured alkane water partition coefficients. Then I'm actually going to get into the subject matter of the actual title, the hydrogen bond donor acceptor asymmetries. Don't get too scared by the term asymmetry and stuff. It's just it's a lot of this stuff you'll be maybe familiar with already. I'm going to talk about the design implications of hydrogen bond donor acceptor asymmetries, and I'll finish off with a couple of slides on frustrated hydration, which it's not strictly a hydrogen bond donor acceptor asymmetry, but I think it's kind of related to hydration, to solvation, to and really if you're trying to design or optimize potency against your targets. A lot of this draws on, I've given a couple of references here. I'm making these slides will be available. They actually are already available on Figshare. The links should work. So when you see the little sort of small print at the bottom of the slide, don't worry about it too much. It's really there, really primarily if you want to go back and have a look at the slides later. So I thought we'd start off with what are we actually trying to do when we design drugs? And I'm introduce you to something I call target engagement potential. So I think when you're doing drug design, it makes sense to sort of try to just understand, exactly what you're trying to do in molecular terms. So first design objectives are the affinity-based design objectives. So you obviously want a very high affinity for the target, and you want to have a very low affinity for the anti-targets. So the anti-targets would be things like hERG, the CYPs, the metabolic enzymes. You don't want to normally inhibit them. And so you ideally, you don't want to be able to measure affinity for these. You want want things to be really very low affinity. Then you go to what I call the exposure-based design objective. And really what this means is you want to be able to control the drug concentration at the site of action. So we say the unbound drug concentration. And this is obviously a function of dose, of time, and location in the body. So this is quite difficult well, you can't measure it for starters. Here's some essential reading for the drug designer, and I recommend this to anyone who's interested in drug design or even probe design, that you have a look at this, because it's the intracellular drug concentration. You can't generally measure it in vivo, but it's a very important part of drug action. So I'm going to talk here about property. You'll see a lot about like property-based design, and this basically mainly means controlling lipophilicity, which I'll have got a nice couple of slides on lipophilicity later. So I'll show you kind of, if you're not that familiar with lipophilicity, the slides will sort of help with that. So what we've got here is we've got this green line represents the probability of getting enough affinity against the target. The red line is the probability. This indicates the likelihood of getting acceptable physicochemical properties. Oh, sorry about that. I pressed too hard on the mouse. Yeah, that's. And then when you multiply these two probabilities, you get this sweet spot in the middle. So this sort of looks approximately Gaussian. So this gives you an idea of the sort of probability of getting acceptable affinity and also getting acceptable exposure. And so what we've got here, this is like the same sort of plot here, but what I've done here is I've moved the two lines further apart, so the sweet spot gets smaller. So this is the problem of getting everything right in a single molecule. Here, what happens, the lines are steeper. So again, the sweet spot is less distinct. And so you're really working on a bit of a hair trigger there. So this is very much like the Mike Hann complexity model that some of you may have encountered. There's a link to it if you've not come across it before. What I'd like to sort of give you an idea here is think of drug design, first of all, as an exercise in manipulating polarity. So this means you put the polar atoms in particular places to optimize the interactions with the target. But remember, this all happens in water, so you can't forget about the water molecules. And then you're also trying to control the total polarity of the molecule to balance aqueous and lipid solubility, because you need the aqueous solubility to get it sort of into solution so that it can travel across the gut wall. But if you make it too soluble, it won't go into the lipid bilayer, so it won't be permeable. So this is a real problem in drug design, is this idea of balancing, getting the polarity so that just right. If it's too polar, it will go into water, but it won't be permeable. If it's not particularly, if it's very lipid soluble, it probably won't be aqueous soluble. So here's some views of polarity. We've got the binary view, polar, non-polar, and this is where polar surface area. You might have encountered polar surface area. It's a very popular descriptor. Sometimes it works, sometimes it doesn't. The next level is polarity has sign. So we have numbers of hydrogen bond acceptors and hydrogen bond donors. The hydrogen bond donors and acceptors are seen as different. So in the celebrated Lipinski's rule of 5. And finally, polarity has sign and magnitude. And this is the sort of stuff this is what interests me in particular, is the idea of getting the magnitude right, understanding that. So here I'm going to show you some stuff on hydrogen bonding. Now, one of the things you think, well, if the element is very electronegative, it'll have a large partial negative charge, so it should be a stronger hydrogen bond acceptor. But actually what happens, this is a measure of hydrogen bond basicity. So this is how good the hydrogen bond acceptor is. And this is a measure of electronegativity. And you can see it goes in completely the opposite direction to what you expect. Fluorine, for all it's going to have a larger delta minus partial negative charge, but it's actually a very weak hydrogen bond acceptor. Now, here's another way of looking at hydrogen bond acceptors. What I've got here is I've got, imagine these azenes. So I have triazine, pyrazine, and pyridine, and I'm measuring the electrostatic potential at a point along the what we call the lone pair axis. And you can see what happens. A long way away, the electrostatic potential tends to zero. As you get closer to the nucleus, this pyridine, the electrostatic potential gets more and more negative. But then you get to this point here where this is what we call the Vmin, the minimized electrostatic potential. After that, it tends to go up. So what this picture is here is that really a hydrogen bond acceptor is not like nitrogen with a negative charge on the nucleus, but more like nitrogen with a kind of partial positive charge with this sort of like this lone pair electron density sort of sitting out here. So just something to think about when you're thinking about hydrogen bonding. One of the things as well is water, of course, is a hydrogen bond acceptor. And when you form hydrogen bonds, you perturb the other hydrogen bonding groups in the molecule. So I've drawn the waters with just a single lone pair. And in this case here, I form water dimer. And what you can see is when I form water dimer, what I call the donor water becomes a stronger hydrogen bond acceptor. Look at the length of these arrows, and it becomes a weaker hydrogen bond acceptor. And the acceptor water molecule becomes a stronger donor. And here we've got I'm just going to check one thing. I'm just checking I haven't got disconnected from the internet. Could somebody just let me know?
I can hear you.
Okay, good. Yeah, it's just sometimes the internet can go. So just I just had visions of sort of talking, everyone lost the internet or something. So I'll periodically do an internet check.
Okay. Well, if you disappear or if we stop hearing you, I think I would certainly step in and.
Yeah.
And I think my colleagues can also help.
Okay, that sounds good. So yeah, if I disappear, just let me know. Yeah. Okay, so getting back to here we've got like this N-methylacetamides. This is a pretty strong hydrogen bond acceptor. Here I've drawn both the lone pairs for the oxygen, and you can see what happens. Same thing happens with the water molecule. But what happens is that one of the lone pairs disappears entirely because it's donated to the hydrogen. The hydrogen bond donor is donating hydrogen bond, and the other lone pair becomes smaller. So I promised you some slides on logP. So what I'm going to talk about here is partition coefficients. But I'm also going to show you how, depending on what the organic solvent is, you get different behavior. So here, this is octanol, this is pyridine, octanol and water. You got the piranha here to tell you this is the water layer. If the piranha was up on top, it would be swimming backstroke. So the piranhas don't go into octanol or or hexadecane. So what you can see is that the pyridine, actually between octanol and water, it prefers to be in the octanol. So if you looked at pyridine, you'd say it's lipophilic. In hexadecane and water, it's different. Here, the pyridine prefers to be in the water, so you'd say it's hydrophilic. And so this is what you can do is you can look at the difference in the two log P values. We call that delta log P. And that gives you a measure of the hydrogen bonding capacity of pyridine. One of the things I just really want to flag out is that octanol water is not really a great way of looking at polarity or solvation. And a good way of showing you this is that octanol water logP just doesn't see the hydrogen bond donors. So here we've got a couple of these methyl transfers where you remove the methyl from the heteroatom to a carbon. And in each case, you can see with the alkane water, you can see that the alkane, the partition coefficient decreases quite significantly. But the octanol water, it hardly changes. In fact, here it increases slightly. So this is kind of, now we're going to get on, well, how you can use alkane water logP to quantify polarity. So this is quite an important slide, so spend a little time on it. What I've got here is this is on the y-axis, I'm plotting, this is a measured alkane water logP. So measured alkane water partition coefficient, the logarithm, of course. Here we have molecular surface area. Now this is molecular surface area, not polar surface area. And I want you to look at this line, first of all, which is this is the line for the alkanes. And you can see if you plot the alkane water partition coefficient for alkanes, you see you get a pretty good straight line. Yeah, certainly if I got my models for bioactivity looking that good, I'd be very, very pleased indeed. And these are saturated hydrocarbons. Now, what I'm doing here is these are basically saturated monoethers. So these are like essentially saturated hydrocarbons, in which case I've changed only a single carbon into oxygen. So everything else is saturated hydrocarbon. And you can see this, the lines are pretty much parallel. And what that's telling us is what we can do is we can use this distance here between the lines to tell us that, to effectively quantify the polarity of an oxygen. So you can see there that's, well, I'll show you the figure in the next slide, next couple of slides. Now these are the saturated monoalcohols. So you can see that this distance here is actually greater than this distance here. And what this is telling us is that the ether oxygen is actually it's more polar than the hydroxyl hydrogen. So I'll get onto this in a bit more detail later. And then here what I've done is I've got diols. So you can see that if you look at this distance here from here to here, it's approximately half the distance of this from here to here. So these are the diols. So this is basically how I quantify polarity. I use structurally prototypical model compounds and get those. So I think probably best to go to the next slide now. This is sort of how we can write polarity. So I write the polarity as this is basically the equation for that top line for the alkanes. And so it's simply given, the polarity is given by the alkane water partition coefficient that you would predict for the compound if it were an actual saturated hydrocarbon of that molecular surface area. And then I subtract the actual measured alkane water logP. That's how we get polarity. And then what I do is I try to use this for prediction of that, but you can also use the measurement to derive that. So I will try to write this. This can be written as a sum of basically over the non-hydrogen atoms. So this over the hydrogen bond donors and acceptors. So I won't really be going too much into this particular equation, but I'll show you kind of how this works now. So here we've got polarity values. These are what we call the Q values. So these mean that these are related to individual hydrogen bond donors and acceptors. This is the graphical abstract from my JMed Chem paper. I say the colors are quite intentional. So I was quite pleased with this. One of the actually normally I find myself when writing a paper that the graphical abstract is something I'm scrambling around to do right at the end and wondering what do I do. In this case, the graphical abstract was ready probably about 2 or 3 months before the paper was actually submitted. So this is how it works. What I do is I can use that. If I take, say, a bunch of aliphatic nitriles, I can actually calculate a polarity for the aliphatic nitriles. And if they've not got any other polar atoms in the molecule, I assign the polarity of the molecule to the hydrogen bond acceptor. So you can do this here. And you can see one of the more notable ones is the sulfoxide. You can see there's a very, very strong hydrogen bond acceptor. We know that from hydrogen bond acceptor strength measurements. And we can see this you see it in the alkane water partition coefficients. Now, the problem is when you have a hydrogen bond donor and acceptor in the molecule, you can't really use this approach because here you have to have a single polar atom in the molecule. So what we do here is actually I get this figure here, which I get from tertiary amides. And then I can look at what the figure is, what the molecular polarity is for secondary amides, and I effectively subtract the polarity of the tertiary amide from the polarity of the secondary amide to get the polarity of the hydrogen bond donor. And so you do similar things here with the imidazole. Now, these are a little tricky because these are, I'm assuming here that you generally, when you get a hydroxyl group, if you remember that slide I showed you with the water dimer, you do tend to find that as the hydrogen bond donor donates to maybe a water molecule, the hydrogen bond acceptor becomes stronger. So it's what it means is a portion of the polarity of a hydroxyl group. You can't really assign it to the donor or to the acceptor. So what I've done here is I've arbitrarily loaded that extra polarity onto the hydrogen bond donor. And you can see that even then, the hydrogen bond donor is, even when you load all that polarity onto the donor, you can still see that the hydrogen bond acceptor is more polar than the donor. And the phenol is the one where this is a very strong hydrogen bond donor. And again, you can, from hydrogen bond acidity measurements, you can see how strong, you can see the strength of the phenol. So that's, so finally got onto the asymmetries. 20 minutes into the presentation, you finally get to the subject matter. That's, I guess, sort of par for the course, really. So the asymmetries, first of all, hydrogen bond acceptors are typically more polar than hydrogen bond donors. Now, I've just shown you that in the last slide. I have some other data, but I think that hopefully that slide will convince you of that. What this means is you have to focus on the hydrogen bond acceptors to address polarity limited permeability. Hydrogen bond acceptors are also typically more numerous than hydrogen bond donors in molecular structures of interest in drug design. And so what this means, this tells us if you're trying to break the hydrogen bond, to increase the hydrogen bond acceptor donor imbalance, if you increase that, you will effectively, you will affect the packing, you will destabilize the solid state. So this is what we say, this is probably a tactic for increasing aqueous solubility. The other thing is the presence of a hydrogen bond donor actually should be a molecular structure typically implies that presence of a hydrogen bond acceptor, but not vice versa. And this is primarily relevant when you're optimizing target liquid interactions. And I've got a couple of slides later, we'll show you how that works. hydrogen bond donors actually, here's a sort of, I don't know, I wouldn't call this an asymmetry, but here the hydrogen bond donors are denounced as the enemy of medicinal chemists. So again, here's a literature reference. You can check and see whether you agree with this. But generally, there's a belief in medicinal chemistry that hydrogen bond donors are bad. My view is that really it's not always clear whether there's, how strong the evidence is for that. But that's what people think. So I'm going to talk a little bit now here about the rule of 5. Now, I'm guessing that a lot of people will have heard of the rule of 5. This is Lipinski's rule of 5, it's often called. It was first published in 1997. My view is actually it's not quite as useful. I think it was, well, let me start again. I think it was very useful for raising people's awareness of physical properties, in particular lipophilicity, as a sort of, something you've got to worry about in drug design. But it's not really a design tool. Here's got the reference here. So the Lipinski rules basically says that you're going to have poor absorption or permeation when certain things happen. One of them is that you have more than 5 hydrogen bond donors, and another says you have more than 10 hydrogen bond acceptors. Now, if you look at that, you say, well, actually, maybe this is evidence that hydrogen bond donors are worse because you can only have 5, whereas you can have 10 acceptors. But I'll show you some others. This is where you really got to sort of look at the some of the other stuff. First thing here, what I've done is I've taken the mean. This is the ratio of the number of hydrogen bond donors to the number of hydrogen bond acceptors. Now, in the rule of 5, that ratio is effectively the cutoff. So the ratio is 1 to 2, so half. But actually, if you look at the hydrogen bond donors are, really, if you take, this is Schultz data set, which is a sort of database of drugs, you find that the hydrogen bond donors are sort of fairly scarce. So you could say, actually, that if you take account of the relative abundances of hydrogen bond donors and hydrogen bond acceptors, actually, rule of 5 is actually more permissive of hydrogen bond donors and acceptors. I don't find this sort of analysis particularly useful myself, but I thought I'd show it to you because you've got to be careful, maybe over-interpreting data. This is one I'll just show you down at the bottom, is that the number of hydrogen bond acceptors per polar surface area are very strongly correlated. Now that's polar surface area as opposed to total surface area. And these links should all work, so you can go and check that I've not got a typo or whatever. So we're going to get into sort of design now. Really, polarity limited permeability. It's not always clear that permeability is limited by polarity. It can be quite tough to figure it out. Also, as we see with some of the new modalities, there are some interesting looking structures that are probably our normal sort of limits, cutoffs for polarity are probably not appropriate. And there may be other mechanisms that things are getting into cells. But if you've got your sort of standard, trying to get something into cell and you really believe that you've got evidence that it is too polar, the first thing you need to be thinking about is most of the polarity of a compound of interest in drug design is due to the hydrogen bond acceptors. So getting rid of the hydrogen bond donors isn't really going to help with polarity. You really have to sort of focus on the hydrogen bond acceptors. You could say, well, just put a bunch of, stick a hexyl group or something on the molecular structure. But that's the problem there, is that really is likely to cause other problems. It's also going to, be bad for solubility, and it makes the molecule larger, you get metabolic liability and stuff like that. So really, if you genuinely have polarity limited permeability, my advice is you need to look at the hydrogen bond acceptors. You need to look at can one of them be can you get rid of one of them? Can you make them weaker? Like an ester is a weaker hydrogen bond acceptor than the amide. I've got an example in the hydrogen bond donor paper of people used that in a PROTAC. So that's permeability. the problem with permeability, you can try some stuff. Sometimes things work, sometimes they don't. Now, one of the big problems I'll just mention, because we're talking about permeability, is active efflux is a huge problem when you're trying to get things to get through membranes. What I'll show you here, this is the efflux ratio. So this is a sort of, the idea is that this is, if this number is large, active efflux is a big problem. And what you can see here is this is like what I call this a permeability cliff. All they've done is by moving the nitrogen from here to here, you've effectively eliminated a hydrogen bond donor, and you've kept the rest of the molecule absolutely constant. So in this case here, the hydrogen bond donor is actually bad for permeability. So anyway, that's I've got another example here. Don't worry about that. Again, there's a link. So these examples have been taken from this this article here. So I think the links should all work. This is where I think the hydrogen bond donor-acceptor asymmetries are really probably, I think the advantages are the clearest. And this is where I'd say probably the case for hydrogen bond donors being the enemy is probably stronger than anywhere else. First of all, the hydrogen bond donors, if you eliminate these, when I say non-essential, these mean the hydrogen bond donors that are not actually necessary to achieve binding to the target. Sorry. So what this does is, because you normally have more hydrogen bond acceptors than hydrogen bond donors, if you get rid of one of the donors, you will increase this imbalance. So it means the hydrogen bond acceptors have to work harder to find a hydrogen bond donor when they're forming in the solid state. So what we're saying here is that you can get rid of if you get rid of these non-essential hydrogen bond donors, you've got a possibility that you will get better solubility. The other thing is that fortuitously, because the hydrogen bond donor is also less polar than the hydrogen bond acceptors, if you eliminate a hydrogen bond donor, you keep lipophilicity more under control. So this is fortuitous. It just, if it was the other way around, well, this wouldn't work so well. So how does this work? Well, I didn't have a lot of data. This is actually one from one of my last articles from AstraZeneca, actually. And so what we've done here, I need to get this table a little bit better organized, but hopefully I think you'll be able to see it. What I've got here is I've got these amides, and I've got these are secondary amides. And what I do is I methylate it. Now, what you'd expect if you're thinking about solubility from a perspective of polarity and lipophilicity, you say, well, you've got rid of a hydrogen bond donor, so the molecule is not going to be as well solvated, so it's going to become less soluble. So here, let's go through the first one. So this is an alicyclic, so it's alicyclic amide, and the amine part is an aliphatic, from an aliphatic amine. And what you find is that when you methylate, you get a mean increase in aqueous solubility of about 0.6 log units. The standard deviation is large. This is, again, quite common if you're looking at solubility. The standard error is quite small because you've got a nice sample. And about 3/4 of the time, the solubility increases. So what this tells us, you'll give yourself a better chance of increasing solubility, but it's not guaranteed. This is a much more interesting one. These are acyclic, and these are both the R1 and R2. So these are acyclic benzamides. What happens here is that you get a much larger increase in aqueous solubility from methylation. The standard deviation is actually smaller. I think it's a pretty small sample, and it might be that they're all structurally very related. And the reason for this is you've got this effect here, the same effect as with the aliphatic amides, but you've also got, when you methylate an aromatic amide, an analyde, effectively, what you do, you flip, you invert the cis-trans preference of the amide. So that's, again, you're getting a conformational effect. And so this is the sum of this effect plus the effect that you've flipped the conformational preference of the amide. And so for the 9, I've got 9 pairs here. And basically all of them, you get an increase in aqueous solubility. This is quite interesting here. These are the cyclic amides, and here really there's very little effect. It's slightly positive. It's not within the standard error, so you wouldn't say it's statistically significant. But what is actually happening here, I think, is that with the amides, with these two amides, the acyclic amides, you're getting essentially what we call ladders of infinite ladders of hydrogen bonds, where a carbonyl oxygen accepts a hydrogen bond donor, a hydrogen bond from an amide in another molecule, and then the amide in that other molecule accepts a hydrogen bond from yet another molecule, another amide group. So what this means is you're getting this sort of this hydrogen bond network going right through the crystal structure. Here, what probably happens is you're getting dimer formation. So you're getting hydrogen bonds, but they're just you don't get this sort of long range, sort of the ladders of hydrogen bonds going through the solids, through the crystal lattice. So I think quite interesting, and this also illustrates the importance of, when you do matched molecular pair analysis, the importance of what we call substructural context. You need to look at, what, not just it's an amide, but you need to say, well, what sort of amide is it? And just as a reference, I replaced the aromatic nitrogen with CH. So here again, we're getting rid of a hydrogen bond acceptor. And here, this is what you you might expect, that is that the actual getting rid of the hydrogen bond acceptor actually eliminates the hydrogen bond acceptor, effectively leads to a decrease in solubility. And so that works like that. So I think generally I've got some other matched molecular pair analysis in this actual article. So you might want to have a look. I've got some stuff on plasma protein binding and stuff like that. So that's if you're interested in matched molecular pair analysis, this is quite a helpful paper. Now we're going to get on to the last hydrogen bond donor acceptor asymmetry, and this is, I say, the hydrogen bond donors come with baggage. So generally, what I mean by that is, if you've got a hydrogen bond donor, you generally have to have a hydrogen bond. Generally, hydrogen bond acceptor is also present. So you could take, say, something like pyridine nitrogen or a nitrile nitrogen. There, you can get a hydrogen bond acceptor without having a conventional hydrogen bond donor in the molecule. If you've got a hydroxyl group, you've got your hydrogen bond donor, but you've also got this hydrogen bond acceptor. And the problem is you need to sort of, if you bring this into, if this is all lipophilic here, what you're doing is you're bringing in a hydrogen bond acceptor into contact with a hydrophobic surface, and that's not good for energetics. So the problem is when you're using hydrogen bond donors, you need to think about the hydrogen bond acceptor that might be next to it. And the other problem as well is that when this hydrogen bond donor, when you donate the hydrogen bond, this actually makes this hydrogen bond acceptor a better acceptor. So that kind of exacerbates the problem. So whenever, this is pretty much, if you were in a situation where you might be docking something, you need to think about, not just, you can't just think about that. You've got to think about that as well. And you don't have the same problem you can generally, if with a hydrogen bond acceptor, if you want to present a hydrogen bond acceptor to a binding site, you can generally find a way of doing it without having to drag a hydrogen bond donor in. And then the other problem, of course, is the hydrogen bond acceptors are actually more polar than the hydrogen bond donors. So it's generally, if you happen to have a hydrogen bond acceptor, that you were trying to present, even if it had an attached hydrogen bond donor, that would be less of a problem. So that's something to think about. So that's the last hydrogen bond donor-acceptor symmetry, that the hydrogen bond donor, presence of a hydrogen bond donor generally implies that you've got a hydrogen bond acceptor also present. So now I'm going to finish off with frustrated hydration. Now, this is not quite, this is a slightly off topic from the title point of view, but I think this is kind of fun, and it actually follows on pretty nicely, I think, from the last slide. So I've got it in, so hopefully you'll like it. What we've got here is that when you get two hydrogen bond donors or two hydrogen bond acceptors or hydrogen bond donors aligned, what you find is the water molecules don't really like to see each other. So you get this repulsion, and you get this repulsion here. So this means the hydration is solvated. So this would normally be a pretty good lone pair, and that would also be a pretty good lone pair. These are both individually a good hydrogen bond acceptors, but if you stick them in water and you ask them to simultaneously interact with water molecules, you can see problems. But this is actually should be seen as an advantage. This is an opportunity, because obviously we're always thinking about displacing water molecules. So anything that makes a water molecule easier to displace is a good thing. So now look what happens when they all come together. So although these water molecules you can't form good hydrogen bonds to two water molecules simultaneously because the water molecules repel. If you have the hydrogen bond donors, acceptors here, the hydrogen bond donors held rigidly in place, they are repelling each other. These hydrogen bond donors and these hydrogen bond acceptors probably are repelling each other, but they can't do anything about it because they're locked in position. They're locked in place by the covalent geometry. And so this is, if you've got a situation like this is precisely what you need in order to sort of maximize the binding affinity. So I think this is something that, particularly if one's thinking about simulating and doing simulations and sort of trying to assess individual water molecules, I think this is something that you need to get a handle on. So what we've got here, these are the hydrogen bonds. This is a repulsive interaction. This is what we call an attractive secondary electrostatic interaction. So this idea was introduced by Bill Jorgensen and one of his co-workers back, I think, 1989 or 1990. So this has been around a while. And so what we're saying, now my view is in this case, the secondary electrostatic interaction is beneficial. But I still think probably the major factor is probably the frustrated hydration. It's very difficult when you're trying to assess it's very difficult to assess the different contributions of different factors to affinity. And maybe that's a whole different talk. But so let's see how this works though. So this is going back to my days at AstraZeneca. This was actually my very last AstraZeneca paper. In fact, I did the proofs, actually. By that time, I'd escaped from AstraZeneca, and I sent in the final proofs from a hotel in Rio de Janeiro. So I was just about heading off around the world for a year after I escaped. So this is, here what we've got these cathepsin inhibitors. And I've got basically, I want you to look at X is different heterocycles. So you can see here when X is just pyridine, none of them are that particularly potent. When you put a nitrogen here next to here, it doesn't have a huge effect on cathepsin L. This is unfortunate because this is our primary target. But it actually works pretty well for cat L2. This is actually very bad because this is not just a selectivity problem. This is actually a known anti-target, so you definitely don't want to hit this. And it works wonderfully for cathepsin S as well. So it's actually, for cathepsin S, this would be quite a useful tactic. Now, what this means is down in the S2 subsite, we've got these two exposed hydrogen bond donors, and they can, what we're saying is we get something pretty much like the last slide, although I've done the last slide for 1,8 napthyridine rather than sort of pyridazine. And it's described here. One thing I'll say about this paper is in those days, I wasn't really talking about frustrated hydration, and we're talking much more about secondary electrostatic interactions. I would say these were the secondary electrostatic interactions. What was I going to say? Yeah, so I think one moves on. You know, I think I'd be very disappointed if I was thinking about things in exactly the same way as I was 14 years ago. So, but just a warning that perhaps this paper is sort of really, you know, I'm thinking rather differently now. But I think it's certainly the SAR certainly stands. And I think this is pretty interesting and sort of shows how you can use the frustrated hydration. So finally, I've just to think about, I've talked about hydrogen bond donor-acceptor asymmetries, and I say, first of all, they define optimization tactics. So it tells you that you need to, if you've got polarity limited permeability, you need to look at the hydrogen bond acceptors. Don't go sticking hexyl groups on and stuff like that. And eliminate the non-essential hydrogen bond donors as a tactic for improving aqueous solubility. What I will say generally is sometimes these tactics work, sometimes they don't work. There's sort of no panaceas in drug design. I also showed you how hydrogen bond donor acceptor asymmetries kind of impose constraints on design. That's when I showed you with the alcohol, the hydroxyl group having baggage. So I think you normally need to consider the ligand hydrogen bond acceptors when you're attempting to present hydrogen bond donors to the binding site. So I think that's pretty much all I've got to say. Say thank you all for coming along and joining me today. Thank you very much for your attention. I've really enjoyed coming to talk to you. I'm obviously very happy to take some questions as well. So it's